Welcome back. This is video 3 from chapter 26 notes over nutrition and metabolism. In this series, we're going to look at ATP production, and we're going to look at it specifically from carbohydrate sources. So to begin here, recall that ATP is an important source of energy for the body, and it's got to be constantly synthesized. The primary method of ATP production is through glucose catabolism or breakdown. Now we are going to observe how amino acids and fat products such as glycerol can be converted into components of these different pathways like glycolysis and aerobic respiration. But we are going to start with just an understanding here of how glucose runs through this process. There's 3 principal pathways we will be discussing. The first is glycolysis. This is an anaerobic splitting of a glucose molecule into 2 pyruvic acid molecules. The second is anaerobic fermentation, and this is the reduction of a pyruvic acid, see above, formed from glycolysis, and that pyruvic acid will be formed into lactic acid. And finally, we have aerobic respiration, also known as cellular respiration. And this uses glycolysis, and it takes its product, pyruvic acid, then to oxidize it in the presence of oxygen, so it forms carbon dioxide and water. Now, at each stage here, you can actually see ATP production occur. And so it's going to be important that as we go through, you learn to see how much ATP is formed and how it's formed. And so that's what we look at in this slide here. There's 2 major mechanisms by which an ATP molecule can actually be made. The first is through substrate level phosphorylation, and that's what we're going to see in these first few steps. What's happening here is you actually have an enzymatically catalyzed reaction where you have a phosphate group which is originally attached to some other substrate. And this is a pretty wide variety of substrates or reagents that we're going to look at. And what happens is that phosphate group is pulled off of the substrate and then is going to be passed to an existing adenosine diphosphate molecule. So following this reaction, your substrate now is without its phosphate group, whereas the ADP has now been converted to ATP. Okay, so it's basically a 1-to-1 ratio here of substrate to ATP product. Then you have oxidative phosphorylation. And oxidative phosphorylation for us is going to couple the movement of substances across a mitochondrial membrane to generate ATP. We're going to see a really complex setup within the mitochondria of our cells that allow proton pumps, which are there within the membrane, to actually generate a proton gradient. And that proton gradient is going to power an enzyme here known as the ATP synthase, and that's what's going to generate a large number of ATP. This actually has a bigger bang for your buck, if you will, than substrate level phosphorylation, but we're only going to see it at one step in this process. And so, of course, as you go through and you're looking at those 3 pathways, glycolysis, anaerobic fermentation, and aerobic respiration, it's important to see the similarities and the differences between each of them. To begin with here, we talked about the fact that both glycolysis and anaerobic fermentation are anaerobic, and that means they don't require oxygen. Aerobic respiration will be the only step that actually does, and it's going to require oxygen at the very end of the process with the oxidative phosphorylation component. There's also differences in where these will occur in the cell. Both glycolysis and anaerobic fermentation will occur in the cytosol, whereas aerobic respiration will have components which occur within the mitochondria. With glycolysis, it's going to break down glucose into 2 ATP net and 2 pyruvic acid. Anaerobic fermentation will take pyruvic acid and convert it into lactic acid, generating ATP in the process. And of course, with aerobic respiration, this is going to begin with the pyruvic acid from glycolysis, and it's going to generate carbon dioxide and water and generate somewhere around 30 to 34 ATP. I like this image even though it's not from your textbook because it summarizes the steps of aerobic respiration. Now the reason I bring it up is because the first thing we look at here is going to be glycolysis. So note, glycolysis can occur on its own or it can occur as a part of aerobic respiration. So again, as a summary here, glucose was going to be converted to pyruvic acid, and you're going to see a net of 2 ATP formed in this process. So another thing to pay attention to as we go through is that there's a number of coenzymes we're going to see utilized through ATP production. And this is going to be these coenzymes that we'll see quite a bit in the oxidative phosphorylation component. They are going to rely on the ability to essentially oxidize a number of of products that have been generated throughout glycolysis and anaerobic fermentation. And so what they're going to be working with is a back and forth of what are known as oxidation reduction reactions, sometimes just referred to as redox reactions. And these are going to be catalyzed by enzymes known as dehydrogenases, which remove hydrogen atoms, and oxidases right over here. Anytime you essentially oxidize a molecule, you're looking at the fact that that molecule is losing electrons. Whereas when you say a molecule is reduced, it's going to be gaining electrons. For us, we are going to see a lot of these electrons transported in the form of hydrogen ions. And so quite frequently, when you see oxidized, you're going to see that some molecule is losing hydrogen atoms. Whereas when it's reduced, it quite frequently is gaining hydrogen atoms. Now again, as we said, the enzymes that we're studying here are requiring coenzymes, and these coenzymes are transporting electrons to the electron transport chain. And so the molecules that bring these electrons in the form of hydrogen are going to be known here in their oxidized forms, and this is called the nicotinamide adenine dinucleotide, or NAD positive. FAD is the flavin adenine dinucleotide, and both of these are coming from vitamins that you obtain through your diet. Now we're going to start out here in this more oxidized form, and we're going to see that they're reduced during our early stages of all these ATP generating processes. And again, just to make sure you remember, if they become reduced, it's because they're going to gain electrons, typically in the form of hydrogen. So it's a lot of background, but now we're going to look at each step individually. So again, glycolysis is anaerobic. It's going to be utilizing substrate level phosphorylation. And it begins with a molecule of glucose. Now glucose itself is a 6-carbon molecule. And so as we go through the process, I'm going to spend time referencing this image. As you see here, each of these little gray balls represent 1 carbon atom. And so you would see 6 carbons forming glucose. Now, through this process itself, you're going to see that glucose itself is going to be generated into several different intermediates. First off, this fructose 1,6-bisphosphate, and then that's going to be divided up into 2 molecules, each with 3 carbons, DAP and PGAL, as it's known. And what you see happening here is this. As glucose is converted to this fructose 1,6-bisphosphate, that actually harnesses the energy from 2-ATP. Which is a little bit backwards because the purpose of glycolysis is to generate ATP, and here we are using some. That's important to remember as we go through this process. Following this fructose 1,6-bisphosphate formation, you're going to see it cleave there into DAP. So you can see here this is dihydroxyacetone phosphate, and then over here is glyceraldehyde 3-phosphate. This is the molecule that can also be known as PGAL, and we're going to refer to that quite a bit as we move into the later stages of this PowerPoint. Those 2 molecules that are formed, again, are 3 carbon molecules, and those will essentially be metabolized into what we think of as pyruvic acid. So the 2 precursors to pyruvic acid each have 3 carbons, and so 2 pyruvic acid molecules that you form as products also have 3 carbons each. And during this process, you see that you actually generate 4 ATP molecules. And so when you take a look at the overall product tally for glycolysis, you'll see it written as this. So net products are 2 pyruvic acid molecules and a net production of 2 ATP. So the reason we consider it net production is although you generate 4, you had to use 2 at the very beginning. So it's really only 2 that you make that can be used by other processes. The other thing that you see happening here is you take 2 of those NAD positive coenzymes and you reduce them to form what we call 2-NADH plus H positive. Again, recall, you can see that right here. When they are reduced, it means they are picking up electrons. And that's the idea behind glycolysis. Now, as we continue on, we see this can actually continue into what we know as anaerobic fermentation. And this is what's going to happen in the absence of oxygen. And so again here, you can start to see that those 2 pyruvic acids formed during glycolysis in the absence of oxygen, as you see right here, would pursue this pathway in which they form 2 lactic acid molecules. Right? So again, this is what can happen in pyruvic acid if there's no oxygen present. Now, during this process, you actually will go through and oxidize those NADH plus H positive molecules that you just formed in glycolysis. And this is important because it helps us to recycle those coenzymes. So here they are, newly reduced. And then by actually oxidizing them, you form this NAD positive molecule Right, here we go. That can be recycled and go back here to the initial stages of this reaction with glycolysis. Because the idea here is if you start to run out of this this coenzyme, this oxidized coenzyme, you can no longer allow glycolysis to proceed as needed. And so by recycling that molecule, you allow it to continue until the body no longer needs the process to run. Right? And so right over here, let's see, there we go. Very good. And so over here, as we continue, we see this process occurs in humans when the demand for ATP exceeds the supply of oxygen. And so you're going to see this occurring oftentimes at the very beginning of exercise, where you might not have adequate blood flow to muscles whenever they're starting to harness a lot of ATP for their earlier energy. And maybe they've got a shortage of oxygen because the blood supply isn't where it needs to be. But after a while, after the cardiovascular system picks up and you have dilation of those blood vessels and the heart is beating faster, then you have adequate oxygen and you can move away from anaerobic fermentation. Now, there are cells that do this all the time. For example, you've got erythrocytes, for example, that don't have mitochondria. And so those mitochondria lacking cells, those erythrocytes, this is the only way they can actually continue to generate ATP. Right? So we continue through here just to make sure that this is clear. The lactic acid that is formed is toxic, and large amounts that accumulate can contribute to muscle fatigue and soreness. However, we find that the liver does a really nice job of converting lactic acid back into pyruvic acid whenever oxygen is available again. Likewise, the heart can actually metabolize lactic acid as energy. And so when you hear an individual speak about muscles being sore, it's not usually because lactic acid has built up into too high of levels. The body's doing a good job of metabolizing them. Oftentimes, that's just due to tiny micro tears that you would see in the fascia or in those tendons that attach the muscles themselves. And so back to looking at this idea of how these processes all fit together, what we've looked at again so far is just glycolysis, taking that initial molecule of glucose, making 2 pyruvic acid, and if oxygen's present, then we've made 2 ATP, and we're ready now to roll into this third process, which is cellular respiration. Now, we have talked about these reduced coenzymes, and I'm going to point out on this image where those are showing up. So we took those original NAD coenzymes and we reduced them so that now they're carrying extra electrons in the form of hydrogen. So what you can see here is if we follow the pathway of those reduced coenzymes actually make it to the mitochondria and will contribute to oxidative phosphorylation. And so as we continue to pay attention to the steps of cellular respiration, we are no longer just going to be looking at how much ATP is made, but we're also going to be paying attention to how many of these reduced coenzymes you make. The more reduced coenzymes that you have available, the more efficient oxidative phosphorylation can be. So just keep that in mind, and we'll look at the specifics as we get there. And so to start up, recall that in cellular respiration, you do actually use the byproducts of glycolysis. So the pyruvic acid that you formed here in glycolysis is essentially going to enter the mitochondria, and you can see that right here, and continue cellular respiration. But again, cellular respiration is a process that is actually aerobic. So oxygen is actually required for these processes to occur. And if we're really breaking down what steps are associated with this, you have to have glycolysis. There's also the Krebs cycle, also known as the citric acid cycle, and then what's known as the electron transport chain. You may have seen cellular respiration equation written out like this, C6H12O6. That's actually one molecule of glucose. Remember, it has 6 carbons. You're also going to see 6 molecules of oxygen gas that are fed into this process. And that's going to yield 6 molecules of carbon dioxide, which of course your tissue cells deposit back into the blood and you breathe out eventually, 6 molecules of water, and then ATP. And that's where this equation is so important. And the idea here is that for us, you can use this to keep track of what you're actually generating through each stage as you walk through these steps. Now, one of the benefits here is that even though you're using oxygen, that oxygen allows you to produce far more ATP per single glucose molecule than glycolysis or anaerobic fermentation alone. If you'll recall, glycolysis by itself only generated 2 net ATP, whereas per a single glucose here, you're generating closer to 30. There are 2 different sets of reactions that we're going to look at, and these are matrix reactions, and then of course membrane reactions. The membrane reactions that we look at will occur all along that inner mitochondrial membrane right there with little transmembrane proteins embedded into that, and the matrix reactions themselves happen deeper in this space here. So we begin with the Krebs cycle. Now the Krebs cycle has this initial part called the transitional phase, and then of course we're going to look at the more multifaceted aspect of what is known as the true citric acid cycle. And so in this transitional phase itself, what we see happening is a conversion of pyruvic acid here, right, into what we know of as acetyl-CoA. So we'll look at this process here. It's at the very top of this image, and that shows you the pyruvic acid coming from glycolysis. One thing to keep in mind as you look at this image is this is showing you what happens with one molecule of pyruvic acid. But as you'll remember, in glycolysis, you actually start with 1 glucose and generate 2 pyruvic acid molecules. And so I like to remind students here, right there, that for 1 glucose molecule, this process will occur twice. So keep that in mind as we look at a tally of products that are formed. So you take the 3-carbon pyruvic acid, and from that you remove 1 carbon in the form of carbon dioxide. Again, remember, carbon dioxide is a byproduct here, and you're going to see 6 total per glucose molecule. You also are going to go ahead and reduce more coenzymes in this situation, and then you're going to add a coenzyme A to this molecule to form what's known as acetyl-CoA. And so the goal of the transitional phase is to start with pyruvic acid and make 1 acetyl-CoA. And so as a result, per molecule of glucose, you should see 2 carbon dioxides, 2 reduced coenzymes, and 2 acetyl-CoA formed during this step. Now we want to continue too, because as that acetyl-CoA is formed, it can begin to react with another molecule known as oxaloacetic acid. We call this the pickup molecule because as you can see here, it's a 4-carbon molecule that has the ability to actually bind to this 2-carbon acetyl-CoA, forming citric acid, a 6-carbon molecule. And this is the initial reactant that this citric acid cycle, again the Krebs cycle, is named for. And so as long as you have acetyl-CoA and oxaloacetic acid present, then this cycle can essentially begin. And so here with the Krebs cycle, that acetyl-CoA, as it's combined with oxaloacetic acid, actually loses the coenzyme A, and that can go on to be recycled and added to newly formed molecules of acetyl-CoA in the future. Now, there are a number of steps that this process goes through. I am not going to ask you to know each step, nor am I going to ask you to know the names of all of the intermediates. The names that are important for me in this cycle that you see right here are just acetyl-CoA, oxaloacetic acid, and citric acid. In a nutrition course or a biochemistry course, you would be expected to know all these details. So in addition to knowing the molecules that are involved here, I want you to know what the products are of the cycle itself. And so citric acid, as it's being changed, is going to generate carbon dioxide as carbons are lost. It's also going to be associated with the reduction of more of these NAD coenzymes right there, and is also going to be associated with the reduction of the first FAD coenzyme that we see here. Now, following one cycle, right, you would see 2 carbon dioxides formed, 3 reduced NADH coenzymes, 1 reduced FAD coenzyme, and 1 ATP produced. And so again, if you recall that this is 1/2 of a glucose molecule, then for 1 single glucose, you would see in total 4 molecules of glucose, 6 NADH plus H positive, 2 FADH2, and 2 ATP. And so once again, what we're looking at here is we've run through glycolysis. Pyruvic acid now has gone through the transitional phase to form those 2 acetyl-CoA. Those enter the Krebs cycle, binding to oxaloacetic acid and forming citric acid, and along the way, beginning to generate 2 more ATP, right, per glucose. So 1 glucose gives you 2 ATP, and 6 NADH plus H positives and 2 FADH2. And so we've generated about 4 total ATP so far for 1 glucose molecule. Still not much, but that's why we have to continue to follow these coenzymes all the way here to the electron transport chain. And so as we reach the electron transport chain, we can describe it as just a series of enzyme complexes embedded in the inner mitochondrial membrane. So of course, your mitochondrial matrix is that deeper space within the mitochondrion. Here is the inner mitochondrial membrane, and this is known as the intermembrane space between the inner and the outer mitochondrial membrane. Here are the enzyme complexes we're referring to that are going to make this the electron transport chain. During this process, all these reduced coenzymes right here that we've talked about reach the membrane compounds. And it says here they transfer their hydrogen atoms to these compounds. And this is pretty tricky, as you can see. So remember that the purpose of these hydrogens was to help transport electrons back and forth. And so you can see here that these electrons will essentially be split from the individual hydrogen atoms. The hydrogen atoms actually get pumped into the intermembrane space, whereas the electrons that you see here are actually stripped from that hydrogen atom, and they are transported, right, by these enzymes that you see within that inner mitochondrial membrane. Again, that's what makes this the electron transport chain. So these electrons are also what we would think of as free radicals. I mean, right here in this image, it's nice and clean. You can see them actually being transported from higher to lower energy levels, and then they get picked up essentially and bound into the form of water. So as it looks here, none of them are ever free, as we would think of in terms of a free radical. But of course, the more that this process occurs, there are more of these little electrons that tend to get away and then just float around in space on their own. And that's why you have antioxidants that are recommended in your diet, because those help to actually pick those up. Otherwise, free radicals can cause damage to DNA, and that's a bad thing. Now, as we come back over here and we take a look at these hydrogen protons that we are examining, these form what's known as a proton gradient. So the idea here again is that these enzymes that you see are working as proton pumps. Now, with technical terminology, a pump is going to move some substance from basically a low concentration to a high concentration. So it's moving it against its concentration gradient. So what that looks like for us is we're taking these protons and we're putting them in a very high concentration in this intermembrane space, packing them all together in that space. And as you know, molecules themselves like to dissociate. They like to move from a high to a low concentration. Well, they can't do that until they reach the ATP synthase. And at that point, they can actually move back down their concentration gradient through the ATP synthase enzyme and enter the mitochondrial matrix. Now, as they do that, it actually powers or drives the activity of this ATP synthase enzyme. And it causes the enzyme components to turn or to pivot. And as it does so, that begins to catalyze the formation of ATP. And so once again, as a recap here, you're looking at enzymes which are allowed to split electrons from hydrogen atoms. And those hydrogen atoms begin pumped here into that intermembrane space in a high concentration, and they essentially power the ATP synthase to form ATP as they move down their concentration gradient. And so this is known as the electron transport chain, but the process by which your ATP is actually made is oxidative phosphorylation. Now, I do name the individual enzymes here for you. Complex 1 and 2, coenzyme Q, enzyme complex 3, cytochrome C, enzyme complex 4. But it's not important to me that you actually know their names. Here at the end of this slide is what I do want you to see here. And that, once again, is that it's the oxygen that we breathed in, that we brought in at the very beginning of this process. Again, if we go back and we see this cellular respiration equation, we see that you had to have glucose and you had to have oxygen. Well, this is the reason you have to have oxygen. The oxygen atoms here at the very end are water accepting those what would be free radicals, those 2 electrons, and actually rejoining them with the 2 protons they were separated from to form the water we spoke of earlier. And so here is the oxygen that we are inhaling that's necessary for ATP production. And you can see here that it's actually picking up hydrogens and electrons, and that water as a product here is allowing all these elements to come back together. So again, looking at our cellular respiration equation, you've made 6 CO2 through your transitional phase and your Krebs cycle altogether. And now here are the 6 water molecules you've generated from the original oxygen that was breathed in. And so as you go through here, it's important to know, once again, ATP synthase itself is facilitating formation of ATP through oxidative phosphorylation. You might also see this written as a chemiosmotic mechanism. And the idea there, again, is that that's just referencing the fact that you are allowing protons to flow down their chemical gradient, right? And that actually is what's powering this oxidative process. And so this is just a closer view of ATP synthase, again, highlighting its functions and acknowledging the importance of that proton gradient as it moves down through the enzyme itself, generating ATP. So a couple of review questions here to make sure you can put these elements together. So question 4 is glycolysis can occur in the blank of oxygen. During the reaction, a single molecule of glucose is broken into 2 blank molecules and yields blank molecules of ATP. So find the most correct combination of answer choices here and then enter that in your quiz. The next question, question 5, during the Krebs cycle, and this does not include the transitional phase, but just during the Krebs cycle, metabolism of 1 molecule of glucose will yield blank ATP, blank CO2, and blank NADH plus H positive coenzymes. And lastly here, during the electron transport chain, what is the function of the electrons which have been separated from the hydrogen protons? So choose the best answer. Do they accumulate in the inner membrane space? Do they enter the Krebs cycle as keto acids? Do they pass between enzyme complexes in the inner mitochondrial membrane? Do they drive the activity of the ATPase enzyme? Or do they enter glycolysis to produce ATP? That ends the 3rd video for your exam. You have 1 video left, and so be sure to come back and watch that.